Now we're going to get into another mechanism that can be applied to the neurodegenerative process, but that is also very specific to a type of neurodegenerative disease, otherwise known as prion diseases. So protein misfolding is the general mechanism which we'll be exploring in terms of how that can lead to neurodegeneration in general, but then again, applying it specifically to prion diseases and using prion diseases as a way in which we can explore protein misfolding and how that leads to neurodegeneration within the brain. So, in overview, we're going to focus a lot of this lecture on just describing what proteins are in general, and then looking at how proteins change their structure, go through conformational changes, and become either folded into a active conformer or misfolded into a pathological protein. And that's a really important process for lots of neurodegenerative diseases, such as Alzheimer's, Huntington's, ALS, and Parkinson's disease, and some of the other diseases that we look at, like chronic traumatic encephalopathy after brain injury. So it's those misfolded proteins that then are really essential to prion diseases. So that's where we'll specifically then lecture, or I will specifically lecture, on the different types of prion diseases, how they come about, why they are there, and what is their main pathological mechanism, and what are the clinical symptoms of these prion diseases. So how do they produce changes that are reminiscent, or behavioral changes that are reminiscent of types of diseases like Alzheimer's that lead to dementia, or Parkinson's that also has an aspect of dementia, and then importantly the motor dysfunction. So prion diseases have all of these symptoms that reflect different types of neurodegenerative diseases. And then the histology and the pathology of these prion diseases specifically. So it's a really important distinguishing factor that we can look at in terms of prion diseases to say how they're different from other neurodegenerative diseases that we explore, but really based on the same principle of misfolded proteins. So it's an important common mechanism of neurodegeneration that we should get into in more depth and then apply specifically to prion diseases.
So like I said, translation, we won't spend too much time on the actual process of translation. It's really translating that mature RNA, right, that message into a language or into something that can be used by the cell. So that's a protein, right? It's that, so remember how I likened, DNA is all the information out there on the internet and all these textbooks on neuroscience, and then I transcribe that into these lectures, and then I'm up here translating that information to you, hopefully to understand. Sometimes my translation is probably lost amongst some of you, but it seems to be working out all right. So I'm gonna say that my ability to translate this information into something that you can use is actually successful so far. So we'll see how the next exam goes. But anyway, so the proteome, right, is the full complement of all the proteins expressed by tissues, cells, or organisms at a particular time. And the proteome is made up of these strings of amino acids, right? So what we have here is you have the message that's come out of the nucleus, right? Here's our mature RNA, and now it's the ribosomes come attracted to it. So this is one thing that I'm like, okay, so how does that happen? Why does a ribosome link up with a message? It could be because it's stable, it doesn't have other proteins surrounding it. There's probably a number of different factors, but anyways, the process happens. Ribosomes will locate that floating around message RNA, and then transfer RNA will bring over amino acids. Again, these amino acids are free. They come in from your diet. Like a lot of the essential amino acids are brought in through different foods. And those transfer RNAs will interact with those single amino acids and bring them over to that ribosome mRNA complex and start to build this polypeptide chain, otherwise known as a protein or strings of amino acids. So what are proteins? So now this little thing should start moving pretty soon. So they're polymers of amino acids, right? There we go. So all the amino acids shown in those round colors, and you have your message RNA here. So the transfer RNA picks up a specific amino acid and binds to it. So this is one thing that again, I'll just kind of talk over this while it's running. The transfer RNA with the exact opposite triplicate codon comes close to the ribosome and deposits its amino acid. So then it just starts building this chain of amino acids, and it grows longer, and then it comes out as, kind of a long string, but then it starts to fold upon itself. Okay, so the folded shape or conformation depends on the linear amino acid sequence of the protein. Okay, so what happens is that, right, so it comes out of the ribosome in that linear sequence, and then the way in which those amino acids are stuck together, strung together based on the message RNA, based on the DNA code, is what gives it that, the protein, it's what's called native conformation. Okay, and that's what, I don't like that term too much, but let's just use it for now, and I'll go on with that. So it's the useful shape of the protein. So that native conformation, which I don't think it's really the best way, but it forms the basic to lead to shape changes or conformation changes that then catalyze any reaction or complete whatever function. Okay, so the native conformation provides this base. It's folded, and then that protein can change its shape to produce functions, different functions. But the native conformation, so let me just put this up, and this is why I talk about this. So the native conformation is a protein that's folded into and it forms its lowest energy state. Where's my laser pointer? Okay, so that's this. So basically, when the protein is in its native conformation, it can be very quiet, it can be doesn't require much support or energy requirement for it to do anything. It's in a low energy state. So now we get into, well, how does it become active? So it can become into a higher energy state by unfolding or by interacting with like a phosphate group, an acetyl group, a glyco, a sugar. So it can be altered. It can be post-translationally modified to change its shape. But it can also go into a very high energy state, which is almost misfolded. So what I have up here is the prion protein in its native conformation. So PRP-C is the prion protein, and we all have this in our brains. Every mammal, in fact, I think every mammal that's been studied has this prion protein in its central nervous system. Prion proteins are actually quite functional for all of us. They do they do a variety of things. They're involved in vesicular trafficking. They're probably involved in neurotransmitter release, things like that, real basic functions. Okay, but a prion protein, so it has to go from this native conformation, right, its low energy state, into a bit of a higher energy state to do its function. But sometimes what will happen is that prion protein becomes mutated, or it becomes misfolded, and it gets stuck in these high energy states, and it can't revert back. So what's been discovered is this conformation, this misfolded conformation. So it's a prion protein scrapie. So that SC means scrapie. So it was first discovered in sheep. And what happened is those sheep developed scrapie, or wasting disease in elk, mad cow disease, and in humans it's the prion diseases, so Creutzfeldt-Jakob disease. There's a couple others, but CJD is the one that's most commonly known. And what happens is this misfolded protein, right, becomes stuck in this very high energy state, and it becomes dysfunctional. Okay, and this is like a fundamental property of a number of different neurodegenerative diseases, that these proteins go from their native, normal functioning, and most neurodegenerative diseases are based on a normal protein that's gone wrong or gone wild, I guess is sometimes what people say. It becomes in this misfolded conformation. And you can see that, so if we start thinking about folding native conformation, right, this prion protein in its low native conformation has a lot of these coiled-like structures. Okay, those are absent in the misfolded protein. Okay, and the other one is that you see these longer lines that are in the, or longer ribbons, I guess, that are in the misfolded protein that are not in the native conformation. So they look very, very different. Okay, so this is why I'm going to hammer home this whole idea about protein folding in this lecture and post-translational modification, because the protein, how it's folded, really dictates its normal function, and when it's misfolded, it dictates an abnormal function and often leads to pathology. Okay, so I'm not going to get into how prion disease is spread or anything, but it's really just important at this point to think about the folded shape or the conformation. So people often refer to the native conformation. That's the protein in its perfect form, right? And people love those crystal structures of those proteins in their perfect form. People also love proteins in their, misfolded form, but the native conformation is just this three-dimensional structure that does not require much energy and can interact with other proteins to then turn on or become functional. So you have your native conformation, and then I guess you could say it's unfolded conformation. I would also refer to that as misfolded. Some people call them mutated. I think mutated to me is more of a term for DNA. So I would say that if we can try to stick to proteins as being in their native conformation or a misfolded conformation, that would work well. Or if there's an intermediate, right? There's a kind of a conformation change. And so for me, the native conformation is just fine. That's protein sitting there, waiting to do something. A change is we can say that a conformational change is a positive function of that protein. Okay, so, and then a misfolded conformation is a pathological outcome. Okay, so conformation change is positive, and misfolded is what we'll say is kind of negative, and that says pathological. So here we go. So we have this general structure. So I've already kind of covered a bit of this, but we'll just keep covering it again. So we have the normal protein folded structure, and we'll call that the native conformation. And that can go through conformational changes, right? So you go from this native conformation fold to a different folded structure for that protein. And that can depend most of the time, a normal protein will have these post-translational modifications, PTMs, or interactions with other proteins, and that leads to these conformational changes. So you can also add heat, right, to egg whites. Okay, so if you heat up an egg white, it goes through a conformational change, and then it's done, right? It's very difficult to get it back to that liquid state. And in some cases, this is what people refer to as going into the disease-associated protein or the misfolded structure, right? That you get stuck into this misfolded state, scrambled eggs, you can't go to that, back to that liquid form, and those eggs will become aggregated, and you will lose whatever biological function, or you'll have a gain of toxic activity. So we go to another kind of variety of these conformational changes, is that the conformation change, right, that I put up on the previous slide is normal function of a protein as long as it can go back to its native conformation. So if you phosphorylate a protein like Krebs, right, that's a post-translational modification, then you unphosphorylate it and it goes back to its native conformation and it goes into a low energy state, and that's all good. So, kind of what I said, errors in finding a native conformation due to heat or interference from other molecules can occur. So there's also, in the case of prion diseases, it's a bit weird because a misfolded prion can cause a native conformation prion to misfold and get stuck into this misfolded state. So it's almost like the disease progresses. It spreads throughout the brain by one misfolded protein causing another native conformation unfolded protein to misfold. And then that just keeps propagating that way until there's a loss of biological function or a gain of toxic activity and things shut down. Okay, so if it doesn't go back into that native conformation, right, the result at best is a non-functioning protein. It just sits there and, I mean, it will get chewed up, it will get taken apart. Somehow there could be a chaperone protein that comes and helps it, but oftentimes you can't refold it and you end up with prion-like proteins or Alzheimer's-like plaques, so pathology. So not just Alzheimer's, not just prions, Parkinson's disease, Huntington's disease, a whole host of diseases that are based on proteins getting stuck into these high energy unfolded conformations or misfolded conformations.